Hello everybody, my name is Leilei. I'm from Nanjing Institute of Geography Limnology, Chinese Academy of Science. It's my honor to share my recent research with you. The topic of my presentation is priming effect of cyanobacteria-derived organic matter on steroid estrogen biodegradation in one eutrophic lake. My presentation will include these four parts. First, as we know, the pollution of emerging contaminants, such as antibiotics and estrogens, is becoming more and more serious in aquatic system. These contaminants enter into freshwater through various ways, such as wastewater discharge, sludge disposal, and livestock manure. These compounds pose unprecedented ecological risks to aquatic organisms, and possibly threatening human health through drinking water. For example, exposure to estrogens at concentrations as low as nanogram per liter can alter sexually sexy characteristics, disrupt endocrine function, active hormone responses of aquatic organisms. The common estrogens in the water environment include estrogen, estradiol, estriol, and the most estrogenic compounds, estradiol, which we call EE2. The first three are natural estrogens and easily to be degraded. But EE2 is the main ingredient of oral contraceptive. It is relatively resistant to degradation. Once into the water environment, estrogens will undergo a variety of environmental processes, including photolysis, complexation, biodegradation, adsorption, and biouptake. In the water sediment system of rivers or lakes, estrogens are easy to sink into the sediments and undergo biodegradation by various microbes. However, the environmental concentrations of estrogen are very low. It cannot activate the degradation enzymes in microbes or sustain the growth of degrading microbial taxa. Therefore, the estrogen degradation bacteria are usually multiple substrate utilizers. That's to say, other growth substrates support the growth of potential estrogen-degrading bacteria, and the bacteria then degrade estrogens at very low concentrations. The most important carbon and energy source for bacteria is natural organic matter, we call the norm. Norm is a heterogeneous mixture of complex organic compounds, such as amino acids, proteins, carbohydrates, and humic acids. The main source of norm is autochthonous and allochthonous. The autochthonous norm is mainly derived from the growth and decay of phytoplankton, microphyte, microbes, and zooplankton. This kind of norm is usually biogenic, fresh, and highly bioreactive. In comparison, the allochthonous norm is mainly from the land runoff, rainfall, groundwater, and it is heterogeneous, aged, highly refractive to biodegradation. These two distinct properties may exhibit distinguished effects on the activity and composition of a microbial community. Commonly, the heterogeneous source dominated the norm in freshwater lakes. However, as the increasing eutrophication and climate change in global freshwater lakes, the occurrence of cyanobacteria bloom is becoming more and more frequent, severe. The floating cyanobacteria biomass will release a large amount of norm into water column and then into the sediment. One previous study has revealed that the contributions of autochthonous sources to norm reservoir in sediments of Lake Taihu, China, can be up to 30%. This fresh norm will change the biogeochemical behavior of contaminants in sediments. As compared to other norms, cyanobacteria norm was characterized with a large number of protein, carbohydrate molecules. Our former studies have demonstrated that bioavailability of cyanobacteria norm to microbes was as high as 79%. Among label compounds were mainly composed of proteins, carbohydrates, unsaturated hydrocarbons. The refractive compounds were mainly composed of lignins, condensed aromatics, lipids. As the main substrate, the high bioavailability of cyanobacteria norm must have led to the substrate-controlled succession of bacterial populations, which is significant for the degradation of contaminants. Interestingly, field investigations have found that occurrence of estrogen in Lake Taihu sediments exhibited temporary variation. A lower concentration was observed at summer when the cyanobacteria bloom occurred. Our field investigations also showed that the estrogen biodegradation potential in water column was positively corrected with the intensity of cyanobacteria bloom. It seems that the cyanobacteria norm released may be favorable to the biodegradation of estrogens in sediments. To demonstrate this hypothesis, we sampled cyanobacteria biomass in Lake Taihu during summer and simulated the leaching of norm-formed biomass. To make a comparison, we also sent microphyte to extract the plant-derived norm. Standard humic acid was also used to represent the heterogeneous norm. A 2-month water sediment microcosm incubation was conducted at dark. At day 30 and 60, we sampled the sediments for analysis of EE2, normal mineralization, microbial activity in terms of extracellular polymeric substance production, and enzymes activity. The microbial composition and structure was also analyzed. To understand the composition and the structure of the 3 norm, the absorption and fluorescent spectra were collected. The absorbance of humic acid was the highest, suggested is the highest aromacity and humic degree. However, the humic degree of cyanobacteria norm and microphyte-derived norm was much lower. 5 compounds were obtained through EMPAC analysis. Among C1 and C2 were protein-like compounds, while C3 and C4 and C5 were humic-like components. It should be noted that although C4 and C5 were usually regarded as heterogeneous, but recent studies have reported that it can also be derived from biomass. Cyanobacteria norm mainly included 5% protein-like C1 and C2, and 23% microbial humic-like compounds, while the content of protein-like components in humic acid was very low, suggesting its low bioavailability. The norm mineralization indicated the bioavailability of cyanobacteria norm was equal to microphyte norm, but 3 times humic acid. Significant differences were observed with the effect of norm on carbon mineralization. A novel analysis suggested the primary effect was quantity dependent and quality dependent. For the same kind of norm, the mineralization increased with the norm concentration. EPS is a dominated metabolite of bacterial community. We analyzed the proteins and polysaccharides concentrations in colloidal EPS and bond EPS. Inconsistent with the non-mineralization, the concentration of proteins and polysaccharides in both EPS was significantly increased with the norm input. Moreover, the bond EPS was more sensitive to the norm because it was more dependent on the metabolism of bacteria. The increased EPS was important to sediment stability and self-adjustment, self-protection of a microbial community to exogenous chemicals. With more EPS produced, the bacterial cells were ready to form the community. A complex community including various bacteria could lead to a more close cell-to-cell communication and strong metabolic function. Besides, EPS played a significant role in the chemical binding of contaminants. Hydrophobicity analysis showed that EPS removal will decrease the cellular hydrophobicity. This was not favorable to the trapping of contaminants. The functional groups in EPS can encapsulate more contaminants in spatial proximity to bacterial cells, promote its hydrolysis by extracellular enzymes localized in EPS matrix. The enzyme activity relevant to organic matter metabolism were also analyzed. The three norms mostly presented a positive but temporary effect on the activity of extracellular enzymes. Among the promotion of cyanobacteria norm and the macrophyte norm were stronger. The activity of extracellular enzymes could be regarded as an indicator of the microbial community. Detoxification of exogenous compounds. The increased activity of FDA suggested the strong microbial metabolic activity in norm-amended sediment. DHA is a key enzyme in carbon processing. It transports electrons and hydrogen through a chain of intermediate electron carriers to oxygen. The enhanced non-mineralization could lead to the intense status of respiration. Further Spearman correlation showed that the enhancement of EPS production, enzyme activity, were positively corrected with the C2 content in the 3 norm. But the correlation degree weakens at the day 60. This may be due to the label proteins were depleted. The microbial composition showed different responses to norm amendment. Results of PCOA indicated a temporal variability in the evolution of bacterial community composition. The bacterial communities in same concentrations of cyanobacteria norm and macrophyte norm amended sediments were close to each other. This result highlighted the sensitivity of bacterial community composition to the concentration of alloxonase norm. Moreover, the bacterial community to different concentrations of humic aids amendment were similar, suggesting the roles of recalcitrant substrates in shaping bacterial community were also dependent on its quality. In general, the relative abundance of copious eutrophic taxia were increased in the cyanobacteria norm and macrophyte norm amended groups. At class level, growth of some potential estrogen-degrading bacteria was favorable in the presence of protein-like compounds from cyanobacteria norm and macrophyte norm. Besides the community composition, the community structure was also affected by the norm amendment. We used the network to show the correlation between norm compounds and the microbial OTU. more than half of the correlations were positive. The networks are clustered, modular, and formed a smaller world topology. 5 modules were obtained. Among the proton-like C1 and C2 were divided into one module. The microbial humic-like C3 were classed into one module. The humic-like C4 and C5 were divided into one module. These results suggested the community assemblage were driven by the different norm components. What's more, the dense correlations between the OTUs indicated that the microbial communities had strong interspecific interactions encompassing the biodegradable norm components. The facultative interactions among microbes can promote microbial communities to develop the spontaneous adaptation and microplasticity for contaminant degradation. As expected, the E2 biodegradation with the input of norm was significantly increased. The 1 mg per gram and the 3 mg per gram cyanobacteria norm and microphyte norm both significantly enhanced the E2 biodegradation. However, the 1 mg per gram humic acid did not significantly increase E2 biodegradation, linking the E2 biodegradation with non-mineralization. A positive correlation was found. This simultaneous decrease in TOC and E2 provided strong evidence that co-metabolism dominated E2 biodegradation. So, the cyanobacteria norm increased microbial metabolic activity and reshaped community structure, and then the norm mineralization and E2 biodegradation both increased. According to these results, it can be concluded that the ratio of cyanobacteria norm and terrigenous norm input determined the estrogen biodegradation in sediment. When the cyanobacteria norm was more than terrigenous norm, the high bacterial activity and strong interspecies cooperation resulted in the strong E2 biodegradation. In other words, the cyanobacteria bloom dominated zone may be a hotspot area for estrogen biodegradation, and more attention should be paid to the risks of their metabolites. That's all my presentation. Thank you.